whenever you're ready.
Thank you so much. I'd like to welcome Dr. Yasmin Nasser from the University of Calgary, but before we do so, I'd like to start by acknowledging the ancestral territories of those on whose land we are currently living and working. We anticipate that since this is a UBC Okanagan event, many attendants are in Kelowna on the ancestral and unceded territories of the Silks Okanagan Nation. Many of you are joining us from other regions, so we encourage you to reflect on the lands in which you live and work and who the ancestral caretakers of those lands might be. I also want to thank the UBC Anti-Racism Initiatives Fund, which is funding this particular diversity seminar series, as well as the Department of Biology and the Faculty of Science, and all of the people who are organizing this event. Thank you so much, Robin and Karen and Shirley. It's been an amazing series so far, and I'm really excited to introduce our next speaker, Dr. Yasmin Nasser. She is an assistant professor in the Department of Medicine and a clinical scientist in the Division of Gastroenterology and Hepatology at the University of Calgary. She is a member of the Gastrointestinal Research Group and of the Snyder Institute for Chronic Disease. She completed her MD and her PhD in enteric neurobiology and did her medical residency at the University of British Columbia in gastroenterology fellowship training at the University of Calgary. She went on to complete an advanced fellowship in clinical GI motility and postdoctoral fellowship under the supervision of Dr. Stephen Vanier at Queen's University, where she studied the basic mechanisms of visceral pain and hypersensitivity. She has been the recipient of several prestigious awards, including the Crohn's and Colitis Canada Rising Star Award, a Canadian Association of Gastroenterology paired with CIHR Fellowship, the John Alexander Stewart Fellowship, and a Young Investigator Award from the American Urogastroenterology and Motility Society. Dr. Nasser is a recognized expertise in visceral pain, functional GI disorders, and GI motility, and she runs an IBD chronic pain clinic at the University of Calgary, which is very unique in Canada, and she treats IBD patients that are actually in remission, but continue to suffer from chronic pain. She's also the co-lead for the University of Calgary High-Risk IBD Clinic, which sees undifferentiated patients with diarrhea and abdominal pain with the aim to provide them a rapid diagnosis and early treatment for IBD. She is very well funded, and I'm really excited to hear her talk, and I'm going to hand it over to Dr. Nasser now. Thank you very much for joining us.
Thank you. Thank you to all the organizers for inviting me. Thank you, Deanna, for that very kind introduction. So today I'm going to tell you about some of the work that I do in my laboratory on chronic pain and IBD, and also what I do clinically as well. So these are my conflicts of interest. And these are the objectives I hope to speak to you about today. So we're going to talk about the burden of chronic pain in the absence of inflammation and inflammatory bowel disease. We'll talk about how microbial manipulation modulates the development of visceral pain in a mouse model of IBD. And we'll talk a little bit about some sex differences in key peripheral modulators of visceral pain. So the first thing to note is that the incidence of inflammatory bowel disease, and by that I mean both Crohn's and ulcerative colitis, is actually increasing. So both of these are chronic diseases of intestinal inflammation. Crohn's can affect anywhere from the mouth to the anus. Ulcerative colitis only affects the colon. And these are unfortunately very common. And by 2025, they're going to be affecting 1% of the Canadian population. Now, when we see a patient who has mild Crohn's, like this colon here, or horrifyingly severe ulcerative colitis, like this colon here, we start them on all of our state-of-the-art pharmacologic treatments. And the goal of our treatments is always mucosal healing. And that is what all our treatments have been developed to do. So whether we're thinking about small molecules, whether we're thinking about biologics, the goal of all of our treatments is mucosal healing. But unfortunately, for 20 to 60% of our patients, this is not the whole story. And these patients experience chronic abdominal pain despite being in endoscopic remission. And this is a very devastating outcome for many of our patients. It's associated with a high level of anxiety, depression, increased healthcare utilization, and very poor quality of life. And many of these patients also have widespread somatic pain, so pain that comes from the joints, for example, fibromyalgia-like pain in the absence of inflammation. So this suggests that inflammatory bowel disease is characterized by changes in neurotransmission of pain. But there's really few data surrounding the pathophysiologic mechanisms of chronic pain in the absence of inflammation in IBD. And this has really led to limited effective treatments, such that 5 to 25% of our patients are actually on chronic opiates. So this is a figure from a meta-analysis that was recently published, and you can see that there's about 20% of outpatient IBD outpatient IBD patients who are on chronic opiates. And you can see that fully 1/3 of patients are still given an opiate prescription at discharge from emergency department. And this is in spite of the fact that we know that opiates in IBD are associated with depression, disability, hospitalization, post-op complications, and death. Yet still 1/3 of our patients are still on these. So clearly we need better therapeutics for chronic pain in IBD. Now, chronic pain in IBD, like chronic pain in any other condition, is a disorder of the brain-gut axis. And we know that there's changes in neurotransmission at all these different levels of the axis. So in the brain, we know that our patients experience things like stress, anxiety, and depression, and this does not cause pain, but it changes how pain is perceived at the level of the central nervous system and how painful signals are processed. We know that there's hypersensitivity of these ascending pain pathways and dampening of these descending pain pathways. And we also know that there's changes at the level of the periphery in these patients. And this is all what contributes to chronic pain and symptoms in these patients. Now, I'm primarily going to be concentrating on these ascending pain pathways that transmit painful information from the gut to the central nervous system, as well as what's going on at the level of the periphery with the microbiome. So in order to understand some of the experiments and the data that I'm going to show you, you do need to have a little bit of a brief primer on visceral pain neurotransmission. For the non-neuroscientists in the audience, I am sorry, but I will keep this brief, okay? I promise I will not go crazy. So sensory information from the gut is conveyed to the central nervous system by two different systems. So there's the vagal afferents. They have their cell bodies in the nodose ganglia, and the spinal afferents, who have their cell bodies in the dorsal root ganglia. And as you can see, the colon here is innervated by both thoracolumbar and pelvic, sacropelvic afferents, okay? Now, the same dorsal root ganglion sends about 93% of its axons to somatic structures. So those are, things like the skin, the joints, and only 7% of the axons will go to a visceral structure. So that accounts for some of the phenomenon that we see when we talk about visceral pain. Number one, that it's poorly localized and that it's often referred to another area of the body. Now, if we zoom down to the level of the gut here, we can see that these afferents terminate in every level of the gut wall. But not all of these afferents are nociceptors, or nerves that are involved in the relaying of painful sensation. The ones that are nociceptors are located at the level of the mesentery here and at the level of the mucosa here. And these nociceptors are sensitive to stretch, so mechanonociceptors. And the ones at the level of the mucosa are also chemonociceptors, so they can be stimulated by products coming from inflammatory cells and mucosal and luminal contents. Now, one of the most important ion channels that's involved in nociception are the transient receptor potential ion channel superfamily, which has the TRPV1, TRPV4, and TRPA1 members, among others. Now, the one that I'm primarily going to be talking about is the TRPV1, or the vanilloid receptor. That's the receptor for capsaicin, or the active ingredient in red hot chili peppers. And it transduces mechanosensation and inflammatory injury. The reason TRPV1 and these TRP channels are so interesting is that they can be modulated by neuropeptides and inflammatory mediators that can be released as a result of tissue damage. So cytokines, for example, prostaglandins, and then luminal factors. So that would be products of food or even products from microbes. So one of the concepts to understand here is this concept of sensitization of nociceptors. And this is really important because it can cause changes in pain perception. So patients who have chronic pain, they have painful responses that can become exaggerated, and that's called hyperalgesia. But stimuli that are normally innocuous in these patients can also cause pain, and that's called allodynia. And both allodynia and hyperalgesia occur because of sensitization of nociceptors. And this is just a cartoon showing how sensitization occurs. So here's a TRP channel in the membrane of this neuron. And when we stimulate these TRP channels, there is an increase in intracellular calcium that flows into the nerve terminal. Now, things like inflammatory mediators or luminal factors can act on G protein coupled receptors that are also found in the membrane of these neurons. And then that can lead to phosphorylation of TRP receptors, which can increase their activation. That can lead to increased transcription of TRP receptors and then increased insertion of those into the cell membrane. And that can also lead to modification of sodium and potassium channels. Now, all of this will lead to an increased amount of intracellular calcium flowing into this nerve terminal. And as you remember from neuroscience, that's going to start bringing the cell closer and closer to being more and more depolarized and bringing the cell to threshold. And when the cell comes to threshold, it's going to fire an action potential, okay? And that is going to be sensed by the brain as pain. So you can see that when patients have hyperalgesia, these cells might be firing more and more action potential, and the amount of their threshold will get lower and lower. So these are some of the principles behind sensitization. So what is the role of the microbiome in this whole process? So, lately there's been quite a lot of attention brought to the microbiome. I know you guys have some amazing microbial researchers here at UBC Okanagan. I don't think the microbiome needs too much of an introduction, but there's been quite a lot of data lately showing that microbial products can actually interact with nociceptive nerves. So what data do we have that this can play a role in visceral pain? So one of the pieces of data that we know is that patients who have inflammatory bowel disease, even when they go into endoscopic remission, so you scope them and their bowel looks normal, they have dysbiosis. And dysbiosis is defined as an imbalance in bacterial composition, changes in bacterial metabolic activity, or a change in bacterial distribution within the gut. We also know that TRP receptors seem to interact with microbial products. So these can directly activate TRP receptors. And we know that microbial manipulation, in fact, alters TRPV1 function. We also know that TRPV1 is very important in chronic pain in these patients because a pretty seminal study by Akbar et al. Published in Gut in 2010 showed that these IBD patients with chronic pain who are in endoscopic remission have increased TRPV1 expression on their nerves. And we also know that bacteria can either directly or indirectly stimulate nociceptive nerves through the production of metabolites, for example, short-chain fatty acids. So this is the general hypothesis in my laboratory, that the dysbiotic microbiome via TRP channel modulation plays a causal role in the development of chronic visceral pain in the post-inflammatory state in IBD. So the mouse model that we use in my lab is the post-inflammatory DSS mouse model. This is a model that was developed by my colleague and close collaborator, Dr. Christophe Altier. And in this model, what happens is we give mice dextran sodium sulfate, which is a chemical agent that induces colitis. So we give it to them for 5 days, they get colitis, and then we allow them to recover for 5 weeks. And at this 5-week time point, there's no macroscopic or microscopic evidence of ongoing inflammation. However, these mice have visceral pain, and I'll talk about how we assess that in our mouse model. So this is the experimental paradigm I'm going to be showing you for some of the data coming up. So what we do is we take our mice, we give them water or dextran sodium sulfate, and we allow them to recover. In some animals, we also manipulate the microbiome using broad spectrum antibiotics. And when I say broad spectrum, I mean super broad spectrum. So vancomycin, metronidazole, ampicillin, and neomycin. So pretty comprehensive. And then what we do is we evaluate both somatic pain as well as visceral pain in our mice. So this is how we evaluate somatic pain. So we use the Von Frey hair test to examine mechanical pain. So what we do is we place the animals on a wire bottom cage like you see here. And we take a filament and it pokes the hind paw of the mouse, okay? And it increases the force. And when the mouse feels pain, it withdraws its paw. And then the other type of somatic pain assessment we use is thermal pain. So we put the mouse on a 52-degree controlled hot plate. And when the mouse feels pain, it might lick its paw, it might vocalize, it might jump, all these kind of things. And to assess visceral pain, so pain that comes from the colon, what we do is we use colorectal distension. So this is a pretty clinically relevant model. So what we do is we implant electrodes into the external oblique muscles of the mouse. And then we give the animal a couple days to recover. And then we take a balloon and we insert it into the mouse's colon. And then we inflate that balloon. And when the mouse feels pain, it will splint its abdominal musculature. So what we're actually recording is an EMG of that abdominal musculature splinting. So let's take a look at some of the data now. So these are our animals that we've given DSS to and allowed to recover. So you can see that when we compare animals that got water the whole time, that there is a shift in this visceral motor response, an increase, which is significantly different from control animals. So these post-inflammatory DSS mice have visceral hyperalgesia. So yay, our model is working. But the other interesting thing is these mice also have somatic hyperalgesia. So both on the Von Frey hair test, you can see that you require less force for that mouse to withdraw its paw, and that mouse spends less time on the hot plate. So now when we add in antibiotics, you can see, for example, that the DSS animals that got 2 weeks of antibiotics, they have more visceral hyperalgesia when compared to DSS alone. However, the most striking thing about this graph is that you can see that the control animals who got antibiotics have quite a lot of visceral pain when compared to the control animals who did not get antibiotics. So there's something that the antibiotics are destroying that is essentially modulating pain. So this is suggesting that the microbiome can modulate visceral pain. However, we're not really seeing much of an effect of microbial manipulation on somatic pain. So you can see that when we add antibiotics, it looks pretty much just like the post-inflammatory DSS mouse here. So the next question we asked in the lab is, can we actually transfer pain? So can we take the microbes, give them to another mouse, and transfer the phenotype of pain? So the experimental paradigm here is that we take our mice, we give them DSS or water for 5 days, so we induce colitis, then we allow them to recover. Okay, and then we get their stool and we gavage it into an antibiotic treated mouse. And we use the antibiotics to disrupt the basal microbiome. Okay, and then we perform visceral pain assessments a few weeks later. So when we do that, now we can see the vehicle treated mouse is here. So that's the one that got antibiotics alone. In red is the mouse that got gavaged from the post-inflammatory DSS stool. So you can see that these mice have increase in visceral hyperalgesia compared to vehicle alone. But you can also see that when mice are gavaged with regular control stool, that there's a drop in visceral hyperalgesia. So this suggests that fecal microbial transplant can transfer visceral pain, and or there's something that commensal microbes are making that's dampening down pain. Okay. You can see, however, that somatic pain does not appear to be affected by the fecal transfer, as there's really no difference between the 3 groups. And there's the same for the hot plate. I just didn't show it to keep the slide pretty. Now, the next thing we wanted to do was to take a look at the actual microbiome of the animals. So this is a graph showing you, or a picture, I guess, showing you comparison at the genus level between control and post-inflammatory mice. So the blue symbols are the families or genuses that are increased in the DSS animals, and the red are the genuses or families that are decreased in the DSS animals. And as you can see here, that there's an increase in some of these particularly short chain fatty acid producing genuses like Lactobacillus, Ruminoclostridium. And in particular, we thought this was quite interesting, that there's an increase in these Lactobacillus, these particular 2 genuses, the FCS020 and the NK. And the reason these are quite interesting is that these 2 genuses are actually found to be increased in patients with irritable bowel syndrome. And irritable bowel syndrome is a very common GI disorder. It affects 11% of the population. It's characterized by chronic pain and changes in bowel habits. And in patients with IBS, these particular genuses have been correlated with visceral pain and the severity of pain, as well as the activation of brain regions involved in the processing of painful sensory motor input. So we thought that was quite interesting. Now, as you can see, we're seeing an increase in these short chain fatty acid producing families. So then we took a look with metabolomics to see if we had an increase in short chain fatty acid content in the stool. So as you can see here, there's a significant increase in fecal acetate and butyrate in the post-inflammatory DSS mice when compared to controls. You can see that antibiotics really kind of get rid of a lot of bugs, and there's not a lot of short chain fatty acids around, which is, good because the antibiotics are working. And you can see that when we do the fecal transfer, that there's also an increase in the mice that got stool from the post-inflammatory DSS mouse compared to the control in short chain fatty acid content. So we next wanted to ask ourselves whether short chain fatty acids have anything to do with visceral pain. And the literature is quite controversial on this. There's some studies showing that, for example, enemas of butyrate is actually linked to visceral pain, whereas other studies do not show that. So it's all species dependent. Patients who have irritable bowel syndrome, when they go on something called a low FODMAPs diet, which actually seems to treat chronic pain. And this is also something we can use to treat chronic pain in patients with inflammatory bowel disease who are in remission. These patients have a decrease in their fecal short chain fatty acids, which never actually come back to baseline. So maybe short chain fatty acids have something to do with pain. So the way we tried to answer this question is that we took naive dorsal root ganglia neurons. So these are pain sensing neurons now. They're naive neurons from a naive mouse. And we incubated these cultured neurons with either short chain fatty acids directly or the ultrafiltrate coming from the colon of these animals. So when I say ultrafiltrate, I mean the colonic contents minus the bacteria. So we filter it through a 0.2 micron syringe filter. And then we loaded these neurons with a calcium sensitive dye. And we took a look at what happens when we stimulate that TRPV1 ion channel with its agonist capsaicin. So when we do that, so in panel A, we've got the data from the ultrafiltrate, and in panel B, we have the data from the short chain fatty acids alone. And as you can see, when we incubate these neurons with the ultrafiltrate coming from the post-inflammatory DSS mouse, that we see an increase in the amount of intracellular calcium flowing into those neurons. And this is also mimicked by the incubation of these neurons with these short chain fatty acids, butyrate and propionate. So what we concluded here is that this data suggests that microbial-derived soluble mediators may be modulating nociceptor sensitization in this mouse model. So far what I've shown you is that the microbiome may play a role in the development of chronic pain in IBD patients. Well, that's all very well, but let's bring this back to our patients. So is this mouse model reflective of who actually develops chronic pain? So this is a study from Alexander Ford's group where they looked at who actually develops chronic pain in patients with IBD. And they had 85 patients who developed chronic pain who were in clinical remission, and you could argue maybe mucosal remission. And then they had patients with quiescent IBD and active IBD. And they looked at all the patient characteristics as well as, the type of disease, Crohn's versus colitis, whether there was an impact of the distribution of the disease, whether there was an impact of the drug that they were on. But they found that there was really no baseline differences across the groups. And the only things that predicted whether or not patients developed chronic pain was whether or not they were tobacco users and whether or not they were women. So all the data I've shown you so far was done in male mice. So why does sex and gender matter in pain? And why should we be studying sex and gender? So this is a paper showing that pain neurotransmission is sexually dimorphic. It is a seminal study from Jeff Mogul's group. And I think most of you will probably be familiar with this because the CIHR actually uses this study in their sex and gender training modules. Okay, for those of you who've not read this paper, it is, the original paper is published in Nature Neuroscience. It's 4 pages. It's 2 figures without the supplemental data, and it is mind-blowing, and it will make you excited about science all over again. So this seminal study from Jeff Mogul's group, what they were doing was they were looking at a nerve injury model, and they were interested in what predicted mechanical allodynia in this nerve injury model in mice. And they found that in male mice, that this was microglial dependent. So inhibitors of microglia seem to all block allodynia in male mice, but not in female mice. Whereas in female mice, the mechanism was found to be T-cell dependent. And this is a consensus report from the International Association for the Study of Pain showing that it is really quite important to study both sex and gender differences in pain and analgesia. Now, this is an old consensus guideline. I think this is from 2007, but there's been new ones that have come out since. And some of the things they talked about, so if we're looking in people, that we should consider both sex and gender, and we should consider both clinical as well as psychosocial factors that are relevant to sex and gender influences on pain and analgesia, things like age, ethnicity, psychosocial, psychological comorbidities, and things like a history of physical and sexual abuse. There have been systematic reviews and meta-analysis which have shown that gender stereotypes specific to pain scales really affect how pain is sensed in patients. So, For example, patients who considered themselves more masculine and less sensitive to pain than typical men showed higher pain thresholds and tolerances, while femininity was associated with greater sensitivity to painful stimuli. And we also know that coping strategies are different between genders, so very important in patients to consider both sex and gender. Now, in preclinical models, they did recommend, it is important to compare sexes. And one of the things they did stress and is important to control for is that pain sensitivity can be affected by the estrous cycle. So let's now take a look at this data. What do we know about sex differences in animal models and visceral pain? So these are some data from rat models. Where they showed that visceral hypersensitivity really varies across the estrous cycle. So this is a study from 2008 here, where they looked at the visceral motor reflex to colorectal distension. So that same model of visceral pain that we're using. And they showed that rats who were in proestrus had increased sensitivity to visceral distension of their colon compared to rats who were in metaestrus or diestrus. And then the same group went on to look at the effect of sex hormones on this effect. And here they were interested in looking at both chronic stress and pain. So what they did is they put their rats through a forced swim model. Okay, so rodents don't like to swim. The water is cold. They get grumpy. So what they did in this experiment is they threw the rats in some water. The rats are swimming around, and then they pick them up afterwards. Okay, so that's a stressor that the animal is subjected to. And they found that when they looked at the visceral motor reflex to colorectal distension, that these animals had quite a lot of visceral pain. And in females, this persisted up to 18 days after the stressor. In male mice, they had increased visceral pain up to 2 days, and then they were just back to baseline. And they found that they could reverse this effect in the female mice by ovarectomy, so removing the ovaries, and they could mimic what happened in the female mice in the males by an orchiectomy, so removing the testes. So this argues that sex hormones really play a role in visceral pain. And we know that data from studies in migraine, for example, show that there's really these pain sensing neurons do express receptors for sex hormones. So we know that dorsal root ganglion neurons that sense pain express receptors for the estrogen, so both the sort of classical receptor estrogen receptor beta, as well as these G protein coupled receptors like GPR30. And we know that when these are stimulated by estradiol, you get activation of kinases like PI3 kinases and PKC that can cause phosphorylation of these TRP channels and increase intracellular calcium. We also know that estrogen receptor beta, when it's stimulated, increases the expression of TRPV1, and an increased number of these channels get inserted into the cell membrane. We also know that that results in increased release of neurotransmitters like calcitonin gene-related peptide and substance P at the level of the central nervous system, causing increased central sensitization and pain. Well, what do we know about sex hormones in the microbiome? So this is a seminal study done by Org et al., published in Gut Microbes in 2016, and they looked at sex differences in the microbiome across 89 different strains. I feel really, really sorry for those poor graduate students who did these experiments. And they found, unsurprisingly, that the impact of sex depends on the strain. And the ones that had the most differences in the microbiome between males and females were the C3H-HEJ mice and the Black 6 mice. And then they're showing some of the genuses that are increased in these animals. So females had an increase in lactobacilli at baseline, as well as some short chain fatty acid producing families like ruminococcus, whereas male mice had an increase in some other of these strains. So what do we know about the microbiome and visceral pain? So this is an interesting study that was published by John Cryan's lab, and they were looking in germ-free mice. So these are mice that are born in a germ-free environment. So they have never seen bacteria. They have changes in their nervous system, we know. And they compared these animals to specific pathogen-free sort of normal lab mice, okay? In 2017, they actually looked in male mice, and they found that when they did that, the germ-free males were hypersensitive compared to the specific pathogen-free. But when they looked in female mice, they actually found that the visceral motor reflex was exactly the same in germ-free females and the specific pathogen-free females. And they found that the estrous cycle effects on the visceral motor reflex were actually abolished in the germ-free females. Now, in their hands, they found that ovarectomy actually caused visceral hypersensitivity. There's been many papers that have shown the opposite, but in their hands anyways, ovarectomy caused visceral hypersensitivity. However, the effects of that were abolished in the germ-free females. So this data suggests that the microbiome plays a role in visceral hypersensitivity in female mice. So what do we know about this in our mouse model? So this is the experimental paradigm I'm going to be showing you. And these experiments were all done by one of my very talented graduate students, Maria Arzamendi. So what she's been doing is she's been giving the animals dextran sodium sulfate for 5 days or water, and then she allows them to recover. And then what she does is she synchronizes her females into the same phase of the estrous cycle, and I'll show you how she does that before doing somatic and visceral pain assessment. So this is a slide that I full-on stole from Maria that shows how synchronization is performed. So these are the stages of the mouse estrous cycle. So the way that we've been synchronizing these animals is that we're introducing a vasectomized male to the cycling females. And when you do that, 2 days later, the animals will have what's called a pseudo-pregnancy. So they'll show you a vaginal plug. And then 2 to 3 days later, they will all be in the diestrous phase of their estrous cycle. And so we've been doing both somatic and visceral pain experiments when they're in diestrous. So this is some of the data that Maria has, and this is all preliminary new data. So when she looks at the microbiome and compares males and females who got DSS and were allowed to recover. So on this side of the graph, we've got the families that are increased in the males. And then on this side of the graph, we've got the families that are increased in the females, essentially. And as you can see, in the males, again, there's this increase in these Lactobacillus and Ruminococcus families that I previously showed you. There's also an increase in certain types of short chain fatty acid producing families in the females, but females also show an increase in the Akkermansia, for example. And we know that, what does this mean? So we know that patients with inactive Crohn's disease, for example, have increased relative abundances of Akkermansia, for example, whereas we know that in our pain model in males anyways, that there is an increase in these short chain fatty acid producing firmicutes. And at baseline, sorry, I didn't show you that data, but that there is an increase in lactobacillus in females, which is known. And when Maria looked at the pain responses, now this is data only from the females, you can see that these females that got dextran sodium sulfate and were allowed to recover compared to controls show visceral hypersensitivity. But one of the interesting things here is you can see at 30 millimeters of mercury of distension that there's something called allodynia. So 30 millimeters of mercury is like a really big pellet of poop. So mice shouldn't have pain at that level, but these girls do. So this shows that these female mice exhibit significant allodynia. And, Maria is now looking in overectomized models, and we're going to be doing some immunohistochemistry, looking at what's happening with estrogen receptor beta. So, right now this is very preliminary, and obviously further studies are necessary to understand the role of sex in the microbiome and the pathophysiology of chronic pain in IBD. So let's bring this back to our patients. As you know, I'm a clinician scientist, so the other half of my life is spent in the clinic with patients. And I do run a chronic pain clinic here in Calgary, which is unique in Canada. And I see patients who are in mucosal remission. And in this clinic, what I do is, I ensure that the symptoms that patients are experiencing is not because they have, occult active disease, not because they've got stricturing or fibrostenotic disease or post-operative complications. And then we treat all the areas of the brain-gut axis that can contribute to visceral pain and sensitization. And, some of the things that we do in the clinic is that we treat patients with central directed therapies. So things like cognitive behavioral therapy, which changes cognitive biases. These are psychological techniques to treat pain. We treat them for their PTSD. Many IBD patients have terrible PTSD from many of the medical procedures and surgeries, for example, that they've had. We treat them with pain-directed therapies aimed at changing how visceral pain is transmitted from the gut to the brain. And we also treat them with therapies aimed at modulating the microbiome, for example, dietary-directed therapies. And the other thing we really do is we really phenotype our patients. So it's really important to understand everything that's contributing to pain in our patients. So talking to them about, their psychiatric disease that's concomitant, talking to them about their quality of pain. So whether it's something that's chronic or it waxes and wanes. And, it's important to look at all of these factors in our patients, not just sex and gender, although that is very important, as I've just talked to you, to really get an understanding of their pain endotype. And what, we're hoping is all of this data is going to lead to new therapeutic targets that are based really on the pain endotype of these patients. So taking in all these different factors into consideration. Many of these patients are also very kind. They're very keen on research. And I've been very fortunate to recruit these patients to giving us stool to put in our mouse model. So this is also something that these patients have been really helping me with. And so it's a very rewarding experience. So in conclusion, I hope that we've talked about how chronic pain is a significant burden in patients with inflammatory bowel disease, and that maybe microbial manipulation will provide novel therapies for chronic pain in IBD, either through the use of things like fecal transplant, for example, or agonism or antagonism of particular microbial metabolic pathways. So these are my acknowledgements. These are the past and present members of my lab. The data from the CMGH paper was all performed by my postdoctoral fellow at that time, Nicolas Esquer. Maria Arzamendi is the graduate student right now who's been comparing male and females in the lab. Adam Schutz is a long-suffering postdoctoral fellow in my lab who's really looking at some of the mechanisms in signaling between short-chain fatty acid and neurons. Ron Chan is my research associate. Nothing would get done in the lab without Ron. And of course, our collaborators in the Altier lab, the Sharkey lab, Ian Lewis and his postdoc Dominique Bihon, with whom we do all the metabolomic work. Marcus Goeking, a good friend and collaborator without whom nothing microbial would ever get done in my lab. Alana Schick was one of the outstanding bioinformaticians with the IMC who helped us analyze that data from the CMGH paper. I'm a junior career clinician scientist. I'm very fortunate to have a mentorship committee who has been helping me, consisting of Christoph, Keith, but also Morley, Wally, Derek, and Mark, and of course, the fabulous funding agencies for their support. Thank you very much. I'm happy to answer any questions.
Thank you so much, Dr. Nasser. That was really, really interesting. Are there any questions from the audience? Maybe I'll just start one off. before people have a chance to think about theirs. I was curious about the TRPT receptors, and you mentioned that these are the receptors for spicy food. And I'd really like to know what the literature or what you think. Does spicy food exacerbate pain, or does it actually help? Because I've read different things depending on the evidence seems a bit contradictory, but I'd love to know your perspective on that.
Yeah, so I think, in terms of like when you have repeated stimulation of these receptors, you actually get down regulation of them. So, so for example, and I don't know if this is something that's really been shown clinically and clinically relevant fashion in patients, for example, who eat spicy food. But if you repeatedly stimulate these neurons with capsaicin, you're going to downregulate that receptor. So
that's cool to know. My husband will be really happy about that. Okay, there are a few questions coming through the chat. So one question is do you plan to include control groups of mice given painkillers for the visceral assessment? And if yes, what kind of painkillers would you like to try?
Yeah, so we don't we don't give mice, for example, non-steroidal anti-inflammatories when we're doing the visceral pain. We would give them non-steroidal anti-inflammatories when we implant the electrodes, like when we do the surgery, but they're not anesthetized or have pain modulating agents while we're doing the somatic or visceral pain, because that would obviously dampen what we see.
Okay. Anybody else have questions? Robin?
This might be a little bit of naivety about how this type of research is done, but I noticed you looked at a bunch of different breeds of mice and saw differences. And so it made me wonder if we would expect to see similar types of differences in, I know breeds of mice and races of humans are not exactly the same thing, but there's other differences. Would we expect to see something like that as well, since we're seeing these sex and gender differences?
Yeah, and it's hard to know, right? And I think, ethnicity and that, these kind of things are all part of pain expression. So for sure, we would, probably see differences. I don't know the literature on that super well. Now, we did all of our experiments only in the Black 6 strain. That paper that showed all the differences was one that was published previously. But yeah, there's a lot of literature showing that different strains of mice are very different. So sometimes, when you're lying awake at night, you think, how clinically relevant is this when I've got an inbred strain of mice? Yeah. Hard to know.
Yeah. Okay. Thanks.
I have another question. But there's my graduate student's going to beat me to it. So Jessica's asking, are you also assessing depression and anxiety in addition to pain?
So we are in our patient population, like in my clinic, I do that. In our mouse model, we are actually not doing tests for depression and anxiety. So things like exploration tests, marble burying tests, like all of those things. We don't do those in our model. We're primarily concentrating on pain, but a great question and very relevant.
So one of the things I was going to ask is if with so much of the microbiome literature out there, it's like, a little hand waving, and I guess I would like to pinpoint a little bit further. Like if you had to say, is it a taxa, or is it a consortium, or is it simply metabolites? What is your point of view? Like, where would you lean towards? And I'm thinking about treatment, right? So if we had to treat patients, right, should we be looking for probiotics? Should we be looking for next generation probiotics? Should we be thinking more about metabolites to play around with? What's your perspective on that?
Yeah, I think this is probably all metabolites. And I think that's, because the microbiome, we know it changes. How do I say this? We know that the metabolites are something that's going to be a bit more consistent in terms of looking at and in terms of looking at microbial shifts. And we do know that metabolites like short chain fatty acids, but other ones like secondary bile acids have been linked to visceral pain. We know that the microbiome can make endocannabinoid like substances, which can, potentially change visceral pain. So I think metabolites is sort of the way to go. Now, I could be wrong, but I think that that's the way to go. And I think, some of the data behind probiotics has been very disappointing in pain. I mean, there's some data showing that multi-strain probiotics are, weakly associated with improvement in pain and irritable bowel syndrome. Most people haven't really looked in IBD and remission. But those types of recommendations from the meta-analyses, like if you look at the data, they're not that good. And there's some data showing that probiotics make substances that actually signal to the brain. So they're psychobiotics. So there's a systemic response going on rather than something going on at the level of the gut. So I think this is going to be a metabolite effect or something else the bacteria are producing, for example.
Okay, great. Yes, Robin, please go ahead.
I actually was wondering if you were okay if we sort of switched gears a little bit and went more towards the diversity side.
Yeah.
Not that other people can't ask other questions about your work, because it's really cool. But you're very successful. Your bio it's very clear that you are very successful. And especially for graduate students, when they see someone like that, it's really hard to see the path that you took to get there. So I wondered if you could talk a little bit about, if you're willing, of course.
Yeah, of course.
Talk a little bit about maybe the challenges or the, sorry, my phone is ringing and it's spam. My apologies. The challenges that you faced, or the sort of something a little bit about your journey.
Yeah. So, I was always interested in doing research. Like, I think research was kind of my first love. And then it was sort of late that I decided I wanted to go into med school. But then I knew I always wanted to do both. And then I would say one of the things that I did after when I went as a resident and is that I kept kind of my hand in, and I kept coming back to the lab. And I think part of that is my supervisor, Keith Sharkey, who is my PhD supervisor and is still a wonderful mentor and he's like my dad, right? Keith is amazing. And Keith let me just sort of email him, and I would say, I'm coming back from Vancouver for a month. Can I please come in the lab? Please let me do something, please, please. And he'd be like, okay, sure. Here's a project for you. And so it was, through just staying in the lab and staying in research that I was sort of able to, and that kind of also kept me going on the clinical side when stuff was kind of, you see, terrible things happening to people in medicine. And, sometimes when you're able to think, what, I'm frustrated about this, but the lab is allowing me to ask the questions of, in a very reductionist manner about treatment and things like that. And then, again, when I did my postdoc, I was doing both clinical work and research in the lab. And I would say that the reason I have been successful is because I've had people who have supported me and really good mentors. And I think that that's probably the biggest theme. And most people you talk to, like that is what it is mentors and people who help. So, Keith has been just fabulous. Stephen Banner, my postdoctoral supervisor, is an amazing, amazing person and a clinician scientist also, who taught me kind of, you can't see everybody who's got GI problems. You have to be very, you have to tailor your clinical work to your research interests so that you can be successful as a clinician scientist. And then, having this kind of committee when I was just starting out, because you come and it's like, okay, there's an empty lab, now go. And you're kind of thinking, oh, what do I do now, right? So having this mentorship committee has been super, super instrumental and helpful. And Christoph, who actually is right there on the other side of the wall, is been a fabulous mentor for me. He's an amazing basic scientist. He does a lot of visceral pain work, and he and I collaborate quite a bit. And it's just lovely having him on the other side of the wall when I just sort of run into the office and say, oh my God, you got to see this data. And, it's fabulous having those kind of relationships. So sorry, that was very chatty. But
okay, we have lots of time. So you're welcome to chat about it as much as you want. I say this next thing with no disrespect to your mentors because they sound truly wonderful, but I also noticed that your list of mentors are all men. Yeah.
Yeah. So when I started here, it was interesting. The head of medicine sat with me and he said, it's kind of interesting. Yeah. It's like, you've got a good mentorship committee. They're good people. They're people you've known for years and years and they're helping you out, but they're all men. So so I started talking with another clinician scientist who's here, Sonia Battaglia, who's an endocrinologist, who is also a mom like myself. And that's been actually something that's been also very, very helpful and very, very different. Looking at it from a balancing work-life kind of thing with, a young family, but then also talking about some of the challenges that are unique to moms and unique to women. And I've also been lucky to, have other people as my mentors who I didn't list as being official ones, but people like Laura Targonic in Toronto, she's like a tour de force for anybody who's met her. And she's just been fabulous at giving me lots of advice and help as well.
Amazing. Thank you so much.
Yeah, no worries.
Yeah, I was just going to ask, I agree about Laura. That's actually how I met you, is through the women in IBD and in gastroenterology on that committee in Canada. And I guess what I wanted to ask for my graduate students out there on behalf of them, did you ever have any doubts that you wanted to continue your career in this field? Like, I know it's really hard as a PhD scientist and as a clinician scientist, just make that twice as hard. So did you just, go down this path? Was it a straight path, or was it like, what were your challenges? And did you have serious doubts about that?
I think some of the challenges, now I would say the biggest challenge is balancing clinical responsibilities with the lab and being, present enough in the lab, and I think Maria and Adam from my lab are probably in the background there, and they can probably comment on this better than I can, but, trying to be there and available for them to help them has been a challenge, I think, with a busy clinical life. I'm fortunate now that I have more protected time than I did when I started. When I started, I was 50-50, which was insane. And I'm now 75%. So I'm very fortunate to have that. So that's been one big challenge, I would say. Funding is always something, right? So when you're writing your grant, you're competing, a clinician scientist who has 50% protected time is competing with somebody who has 100% protected time and who's, has got an army of graduate students when you're sort of like, I'm starting out and I don't have a lot of funding. So, that's of course a challenge. I won't say I've never said, oh my God, am I crazy? Why am I doing this? And I just said that to the lab yesterday, actually. But, when they show you data and you're just like, oh my God, that is so exciting. Like, that makes your day. There is no high like the thrill of discovery and seeing cool data. But then I think, when, it's science, so it doesn't work, right? So when you've got that, you go to the clinic, right? And you also have that kind of thing. So you've got people who you're helping and who get better. And when they don't get better, you've got the lab to channel your frustrations and ask yourself questions about why they're not getting better.
Yeah, that sounds very familiar, but swipe mine with clinical and teaching, same but different. I really want to thank you, Dr. Nasser. That was a really, really fantastic talk, and I thank you so much for your time, and I hope you have a great rest of your day. Thank you for joining us.
Thank you so much for having me. It's lovely to see everybody. So,
thank you. Bye-bye.